Hello guys, good morning. Welcome to our channel Niperian Solutions. So today also we are going to discuss some previous year questions which is held in the 2023 in February 2023. So let's discuss one by one all the questions. Let's start with first one, right? So the first question is, which of the following method is used in enzyme preparing according to the property of enzyme, right? So what are the options? That is the thin layer chromatography, gel filtration, dialysis and affinity chromatography, right? So what is the answer over here, right? So first we will discuss what is the like TLC, right? So definitely we are not using for that one. Then gel filtration is nothing but our size exclusion chromatography, that is also known as the gel filtration chromatography. Then dialysis and affinity chromatography. So the correct answer here is the affinity chromatography, right? So for the enzyme preparing according to the property of the enzyme, right? Okay, the next question, which of the following creatine kinase level are higher in children's, right? So these are the different creatine kinases: CKMM, CKBB, CKMM, CKBK, CKMB, CKBB, CKBB and CKBK, right? So these are the different creatine phosphokinase or the creatine kinase enzymes. So they are asking the which are higher in the children's, right? So what is the answer over here? So the answer is our CKMB and CKBB, right? So this one, right? Okay, so we will see what are the creatine phosphokinases, right? So these are the different creatine kinase or the creatine phosphokinase. So there are the three isoenzymes of the creatine phosphokinase: CPK1, CPK2 and CPK3. And the different subunits are: for CPK1 that is BB, for CPK2 that is MB, and CPK3 MM. So what is the BB? That is for the brain. Then MB, that is for the heart, and the MM, that is for the skeletal muscles. So here you can see guys, the estimation of CPK2 MB is the earliest reliable indication of myocardial infection. So that is the first detectable like enzyme, or the like what we can say, that is the marker for the myocardial infarction, right? Here also we are given the different isoenzymes, right? Like lactate dehydrogenase, creatine kinase, alkaline phosphatase and isocitrate dehydrogenase. So that is also very important from the exam point of view, which enzyme having how many isoenzymes, right? So lactate dehydrogenase is having the 5 isoenzymes, creatine kinase having the 3 isoenzymes, just we have discussed, right? CPK 1, 2 and 3. Again alkaline phosphatase having the 6 isoenzymes, and the isocitrate dehydrogenase having the 4 isoenzymes, right? Okay, here are the some important enzymes in the diagnosis of the disease. So means which disease and which enzymes is elevated in the specific disease. So that chart is also very important, right? So here we know amylase for the acute pancreatitis. So accordingly that all are very important one. So here is SGPT, SGOT is also important. Just remember guys, SGPT is for the hepatitis and SGOT for myocardial infarction, right? So and just previously we have seen one another enzyme is there for the myocardial infarction, that is the CPK2, right? So these are the important enzymes and the like the most important diseases, right? Okay, the next one, who discovered the ABO blood group system? Oh, that is very basic question. So who discovered that one? That is the Carl Landsteiner, right? So Carl Landsteiner discovered the ABO blood group system, right? So the next one, which of the following arteries is involved in carrying deoxygenated blood from the right ventricle to the lungs? So what are the options? Pulmonary artery, aorta, that is the like systemic aorta, systemic artery and the lingual artery. So if there is a deoxygenated blood, right, from the right ventricle to the lungs, so if the lungs are involved in somewhere, so means what is the answer over here? That is the our pulmonary artery, right? So pulmonary artery carries the deoxygenated blood from our ventricle to the lungs. So that is question from the HAP, right? Human anatomy and physiology. From the HAP, the CVS is very very important. Cardiovascular system and blood. These topics are very very important from the exam point of view. So please go through it. Okay, the next one, the antigens for RH and ABO blood groups are present on, right? So here are the different blood cells, that is the white blood cells, chromosomes, platelets and red blood cells. ठीक है, अब यहाँ पे क्रोमोसोम तो होगा नहीं, राइट? सो बचे क्या, WBC, प्लेटलेट एंड RBC. तो कहाँ पे होते हैं ये एंटीजेंस RH एंड ABO ब्लड ग्रुप सिस्टम के? दैट इज द RBC, राइट? रेड ब्लड सेल्स, right? Answer is red blood cells. Okay, so the next question, the tag polymerase derived from the thermophilic bacteria Thermus aquaticus is active at, so just they are asking the what is the active temperature of the tag polymerase, right? So what is the answer over here? What is the activation temperature? That is nothing but the 72 degrees Celsius for the tag polymerase, right? And the tag polymerase which is obtained from the Thermus aquaticus, that is also very important. Okay, the next question, polymerase chain reaction is revolutionary method developed by, right? So who invented PCR? The PCR technique is invented by the, right? Options are Pauli Flory, Peter Debye, Gary Mullis and Francis Mark, right? So what is the answer here, guys? Right, what is the answer? That is the Gary Mullis. So Gary Mullis discovered the or the invented the PCR, that is the polymerase chain reaction. That polymerase chain reaction is also very very important topic from this FDA exam point of view. So please go through the PCR definitely, right? Which of the following separation is not possible by using electrophoresis, right? So carbohydrates, nucleic acid, proteins and lipids, right? So what are the answer here? Yes, that are the lipids, right? So lipids are not possible for the separate by using the electrophoresis, because for the electrophoresis there should be the electric charge, because these are separated based on the electric charge. So the next one, which of the following technique separates charged particles using the electric field? So just we have seen, right? What just we have seen? What is the answer over here? That is the electrophoresis, right? So these are the simple questions from the all the from biochemistry, some that all the subjects, right? Okay, the next, which of the following are example of polysaccharide and oligosaccharide respectively, right? So options are cellulose and glycogen, raffinose and stachose, starch and glycogen and stachose, right? So these are the our options. So what is the correct answer over here? So our answer is glycogen and stachose, right? So these are the glycogen is the polysaccharide and the stachose is the oligosaccharide. So the next one, the major buffer system in body fluid is, the options are H2SO4, HSO4 minus, HNO3, NO3 minus, H2CO3, HCO3 minus and H2SO3, HSO3 minus. So that is very basic question, that is a very logical question. So sulfuric acid and nitric acid, definitely these are not present in the body, right? Means sulfuric acid and nitric acid are not dominating buffer system in the body. So these two options has been cancelled, right? Okay, so in again here H2CO3 and HCO3, right? So our answer is the H2CO3 and HCO3. These are the carbonate buffers, right? So carbonate buffers are there. Just a second, guys. Right. So carbonate buffer is majorly present in the our body. So we have seen in our pharmacology in diuretics topics also. So carbonate buffer plays very important role to exchange with sodium and potassium, or that water and any mineral balance. So that's why the carbonate buffer are very important. So these are the very logical questions, right? Okay, so next one, which of the following carbohydrate are present as stored energy in plant cell and animal cell respectively? So they are just asking the which is from the like what is the store energy in the plant cell as well as the animal cell, right? So what is the answer here? So these are very basic question, that is the starch and the glycogen, right? So which one is the plant in the plant cell, that is the starch, and in animal cell that is the glycogen. Okay, the next one, which of the following IR radiation sources emit maximum radiation at 5200 centimeter inverse, right? So the options are nurse glower, mercury arc, global source, and incandescent lamp. So what is the answer here? So our answer is the nurse glower lamp, right? So nurse glower lamp gives the radiation at 5200 centimeter inverse. So actually that 5200 centimeter inverse, that value is totally like it is beyond that our normal fundamental region, or what we can say the general IR spectra, because it is always in the like 4000 to 667, right? So that is our general range for our IR spectroscopy. So that is 4000 centimeter inverse to the 667 centimeter inverse, right? Okay, so the next question, which of the following substances has the highest dielectric constant? So that is also very logical question, right? We have to think about it. Then the options are benzene, CCl4, that is the carbon tetrachloride, aniline, and the chloroform. So the first of all, what is the dielectric constant, right? Means the molecule are having the high dielectric constant, means these molecules are highly polar, right? So that is nothing but the dielectric constant, means that it should have that positive group and the negative group, and they are attracting towards the each other. So that is nothing but the dielectric constant. Simply we can say the polarity, right? So benzene, so we know that is the non-polar. CCl4 also, that is not the polar one. In aniline, right? So what is the answer here? In aniline, why it is polar? Because what is the structure of aniline, right? That is C6H5NH2, right? That benzene ring here and NH2, that is the aniline, right? So the structure of aniline, it contains the amine group, right? It contains the amine group, and that amine group is polar in nature, means it is having the highest dielectric constant among these options, right? Comparatively, in these options, aniline is highly polar. The compound which contain the amine group and acid groups, they are very highly polar in nature. So simple examples are the your amino acids, right? So they are very highly polar in nature. These are having the highest dielectric constant, right? So what is the option here? What is the answer over here? That is nothing but the our aniline, right? Okay, the next one, which of the following bases has no keto group in nucleic acid, right? So guanine, uracil, cytosine, and adenine, right? Okay, so what is the answer over here? So our answer is adenine, right? So adenine does not contain the keto group. So that is also very important from the biochemistry. The topic is the nucleic acid. So you have to know that all the structures of your basic purine and pyrimidine bases. Okay, we will see that all the structures here. So there are two purine bases, that is adenine and this one, adenine and guanine. So these two are the purine bases, right? And these another three are the pyrimidine bases, right? So two purine and three pyrimidine bases. So these names are also very important, right? So that 6 amino purine for the adenine. Why it is 6 amino purine? Because the numbering start from this nitrogen, right? This nitrogen 1, then this second one 2, that is the third nitrogen 3, this is fourth, fifth, and this is sixth. So the sixth position is the amino group, right? So that's why it is the 6 amino, and this basic moiety right? Which one? This basic moiety is from the purine, right? So that is the basic is the purine, because these two are the purine bases, adenine and guanine. So basic moiety is purine. Like the similarly in guanine, that is the 2 amino 6 oxypurine. So numbering start from the first NH, then second one is the amino, then third nitrogen, fourth, fifth and sixth is the oxygen. So 2 amino 6 oxypurine is nothing but the guanine. In cytosine, that is the pyrimidine base, right? Cytosine, thymine and uracil. You have to remember these three bases are from the pyrimidine category. So the basic moiety is the pyrimidine, right? So here her numbering is like, here is the numbering is the change. So here is numbering starting from the NH, this NH, this one. So 1, 2, 3, 4, right? So 2 oxo, 4 amino purine, right? So just concentrate on the numbering of the purine and pyrimidines, right? So similarly in thymine also, in thymine 2 oxo or 2,4 dioxy 5 methyl pyrimidine, right? So in thymine there is a methyl group, right? So that is very important. So that's why name is the thymine, that is methyl, right? So is there. And in uracil, that is the 2,4 dioxypyrimidine. So there is no methyl group. So these are very important basic structure of your purine and pyrimidine bases, or the nitrogen bases that we can say, right? So the structure is very important. So what is our previous question? The question is that the which does not contain the like keto group, right? Or which simply it means that the which of the base is like which does not having the oxygen, right? Simple. So what is the answer? That is nothing but the our adenine, right? It does not contain any keto group, right? So sometimes they are asking which of the following like nitrogen base contain the methyl group. So that is the thymine, right? So these are the questions. Sometimes they are asking the names 2 amino 6 purines, 6 amino purines. So you have to remember that all the structures accordingly, right? Okay. So I hope that structure and everything is clear to you guys. And the numbering, yes, the numbering is very important from the exam point of view. That numbering of the adenine, or basically the numbering of purine and pyrimidine, right? So you have to know what are the numbering and start from where. Okay. So the next one. How many hydrogen bonds are present between adenine and thymine? So that is also very basic, right? So, okay. So adenine and thymine. Here. So adenine and thymine. So there are the two hydrogen bonds present in the adenine and thymine, right? So what is the answer over here? 2. What about cytosine and that right, in between C and G, right? So that is the triple bond, right? Means 3 hydrogen bond present in the C and G and 2 hydrogen bonds present in the adenine and thymine. Okay, so next one, which of following is an amine? Estrogen, androgen, winstrol and epinephrine. So the name itself indicate that epinephrine is the amine, right? Epinephrine. What is the epinephrine? Sometimes we can call it as the adrenaline, or that adrenaline nor adrenaline, from this category that epinephrine is there, right? Which of the following is an iodinated derivative of tyrosine? That is also again very simple question. The options are adrenaline, thyroxine, cytosine, insulin. So what is the answer here? So that is the thyroxine. So what is the thyroxine? Right? We have seen in the pharmacology, thyroxine, T4 hormone, right? So T4 and T3, these both are the derivative of the tyrosine. So that is the reaction, iodination and that all the things. So you have to remember. So from thyroxine and like T4 and T3, these are the derived from the tyrosine amino acid, right? Okay, the next, which of the following steroid hormones control the level of excretion of water and salt by the kidney? Just we have seen, right? Right. So what are the options? Testosterone, adrenaline, thyroxine, and mineralocorticoid, right? So what is the answer here? That is the mineralocorticoid. They are used in the excretion of water and salt balance in the body, right? Okay, the next one. Which of the following test is useful for the distinguish between the monosaccharide and disaccharides? Right. So very simple question. बारफोर्ड टेस्ट, सैपोनिफिकेशन टेस्ट, मेल्टिंग टेस्ट एंड गैब्रियल टेस्ट, राइट? तो आपने पढ़ा होगा इन कार्बोहाइड्रेट्स फ्रॉम द बायोकेमिस्ट्री. द बारफोर्ड टेस्ट इज यूज्ड फॉर द डिस्टिंग्विश बिटवीन द monosaccharides and the disaccharides, right? So the next, what type of transition is possible in azo compounds, right? So sigma to sigma star, n to pi star, pi to pi star, sigma to pi, right? So what is the answer here? The answer is pi to pi star, right? So why pi to pi star transition? Because in azo, what is the azo compound like, right? So due to this double bond, it is the pi to pi star transitions in the azo compounds, like so. Or these are the just N2 compounds, like what are the azo? These are azo compounds, okay? Which of the following solvents has the highest UV cutoff area, right? So the options are acetonitrile, water, methanol and chloroform, right? So which of the having the highest cutoff area in UV? That is the chloroform, right? Because the acetonitrile, water, methanol, these are very polar solvents, right? So chloroform is the answer here. What are the isoelectric point of lysine and proline respectively? So these are very direct question from the biochemistry. What is the isoelectric point of the specific amino acids? So you have to go through this, guys, right? So what are the options? 5.6 and 4.9, 9.47 and 6.3, 3.5 and 9.5, 4.9 and 2.3, right? So if suppose if you do not know the isoelectric pH of the specific amino acid, but logically if you are thinking the lysine is a basic amino acid, right? What is the lysine? Lysine is the basic amino acid. So accordingly, so that isoelectric point of that basic amino acid is like within the range of that base pH, basic pH, right? So here are, what is the answer? So the answer is 9.47 and 6.3 is the isoelectric pH of the lysine and proline respectively, right? Isoelectric point is a value of, options are molarity, gram molecular weight, exact pH value and normality. So just we have seen what is the isoelectric point? That is nothing but the our pH, right? What is isoelectric point? That is nothing but the pH value. So on what pH value that specific amino acid or the compound will get in the deuterium ion form. So that is nothing but the isoelectric point of a specific amino acid or any specific compound. Okay, the next one, which of the following type of chromatography is used for the separating compound that have the hydrophobic moieties, right? Right, hydrophobic means non-polar compounds, right? So normal phase chromatography, ion exchange chromatography, gel permeation chromatography, right? So what is the answer here? Yes, answer is the reverse phase chromatography. So for hydrophobic compound separation we are using the reversed phase chromatography. Okay, next, which of the following statement is incorrect with respect to the gas chromatography, right? So they are asking the incorrect statement, right? Okay, let's see one by one. In gas chromatography, the sample to be tested must be volatile. Okay, so that is correct. In gas chromatography, mobile phase could be a gas. So obviously in GC, so what is the mobile phase? That is the gas. In gas chromatography is done at low temperature. No. Gas chromatography. Okay, and let's see the fourth option. In gas chromatography, the stationary phase is a low volatile liquid adsorbed on a solid support. That is also correct. Means what is the answer here? Our answer is gas chromatography is done at low temperature, because they are asking the incorrect one. So gas chromatography is not done at low temperature, right? Okay. So the next one, which of the following non-aqueous solvent is an example of protophilic solvent, right? So the options are ketone, formic acid, chloroform and acetonitrile. So what is the example of protophilic solvent over here? That is the, what is the answer here, right? That is the ketone. Ketones are the example of the protophilic solvents. Next one, who proposed the double helical structure of DNA? That is also very simple one. So that is the Watson and Crick. So in option, Joseph John Thomson, Francis Crick, Galileo Galilei and Charles Darwin. So the our correct option is the Francis Crick. So who discovered the double helical structure? Right, Francis Crick. Okay, the next one, which of the following bases contain an amino acid group at C4? Okay, again the same question from the that nitrogen bases present in our DNA and RNA, right? So the options are adenine, guanine, cytosine and thymine, right? So what is the question? Which of the following bases contain amino acid group at C4 position, right? So what is the answer here? That is the cytosine, right? Cytosine. Okay, in cytosine, that is the fourth position, there is a amino group, right? In, we will see, here is the first number, then second number, third number and the fourth number. In pyrimidine, right, we have seen that in pyrimidine the number is starting from this NH group 1 2 3 4, right? So means 2 oxo 4 amino, right? So that's why our answer is cytosine, right? So this structure are very important, right? So all the purine and pyrimidine bases. So from the biochemistry point of view, you have to go through this topic, nucleic acid, amino acids, specifically like enzymes. So these three topics are very important from the biochemistry. So all the structures, what are the enzymes, elevated enzymes, disease related, or the metabolism of that carbohydrate amino acids. So these are very important topics. So you have to guys go through all the important portions, because very few days are remaining for the exam, right? So you have to go that all the important stuff from the biochemistry. So relatively from all the subjects, like in spectroscopy also you have to go through the UV, NMR, IR, mass. What are the problems, small problems related to the like UV, in IR also what are the frequency ranges. So we will take that spectroscopy, that at least UV, NMR we have already covered, and so in another topics like we will cover UV, IR, mass is also very important. So these three spectroscopy are remaining, so that we will cover one by one. And from again instrumental chemistry, you have to go through that all the chromatography in very detail, right? Because they are asking what is the principle behind this, what are the like stationary phases, what is the normal phase, what is the reverse phase. So these all the chromatographies are very important from the exam point of view. Then again you have to go for all like voltammetry, and then what are the polarography, amperometry. So these are also very important topics. Again in the titration also from the analysis, like acid base titration, complexometric titration. So just you have to go through once, you have to complete whole syllabus, and then you have to revise that one, right? At least very important topics, you cannot skip the important topics from the all the technical parts, because that is the very like important area. There are 40 questions from the technical part, so 80 marks are there, right? So you cannot skip, you cannot miss any single important point from the syllabus, right? Okay, So thank you so much guys. So in the next video we will see the another topic in detail. So up till, thank you, and please subscribe the channel, share with your friends. Thank you so much.